First and foremost, I would like to thanks to INBIOSIS for inviting me. A special thanks to my senior, actually, Dr. Azlan from the same university. Today I was given an opportunity to share with you guys on very important topics, which I believe the topic that I suggested for today's presentation is on the probiotic in oral health, the way forward. So the reason I choose this topic is that previously when we talk about probiotics, it's all about gut probiotics. We want to make sure that our gut is good, and many studies has been shown that there is a link between the brain and the gut. Now we look into another perspective, looking into consideration if there is also the same thing happens with the oral cavity. So that is the idea which I am going to share with you guys. Hopefully it will be beneficial for all the members. All right. So this will be the outline of my presentation. I'm going to talk about the three things. Okay. The objective, my real objective coming here definitely is to visit my friend. The second objective is that I want to extend my collaboration with you members. The reason is that it is no longer working in silo, particularly for us at IIUM, International Islamic University. We are not an RU university and we don't have KPI. So when there is no KPI in terms of publication, therefore it's a bit difficult for us to move forward. So this is the platform that I would say it's good not only for IIUM itself, also for my Kulliyyah, to extend our invitation for collaboration. So hopefully if there is anything from INBIOSIS, then you can bring it at my faculty, Kulliyyah of Dentistry. So that will be the first thing I'm going to discuss about. And number two is about the research and development in regards to the probiotics, prebiotics, symbiotics, and postbiotics. And last but not least, I'm also going to touch a little bit on a commercialization perspective in regards to the probiotic research. Right, so this is my university. It is located, we have various campuses. So the one that very common to us is in Gombang. So, and then normally when people look at IIUM, it is all about Al-Quran and Sunnah. It is all about law. It's nothing to do with science. Yes, we have 6 faculties in Kuantan campus, which is about 3 hours driving, 3.5 hours driving from here. So I drove this morning, Alhamdulillah, arrived at UKM before Jumu'ah prayer, managed to pray Jumu'ah, and then immediately come to here. So it's about 3.5 hours driving. So I would like to invite all of you, if you are interested to do some collaboration with IIUM, it's just 3 hours driving. So some of you drive like a couple of hours, right, every day. So for us, 3 hours to go to INBIOSIS, it's I think it's reasonable. Okay, so this is Kuantan. We are here at International Islamic University, Malaysia. So it's a very close nearby to the beach. We have beach, and then behind the university, if you can see all these, the green thingy. So we have what I call waterfalls behind our university. So if you are interested to do, to enjoy, have some fun at the university, you can just come to our university. Then you can have laboratories. We have some laboratories, even though it is not as good as INBIOSIS, but we are open for everyone to join us. To collaborate with us, right? And then this is our faculty. This is our university. So we have 6 faculties altogether in the in Kuantan campus. So we have Faculty of Dentistry, we have Pharmacy, so we have also Faculty of Medicine. What else? Pharmacy. We have Kulliyyah of Nursing. We have Kulliyyah of Science. Kulliyyah of Medicine is the most important one, definitely. So we have all 6 kuliyyahs altogether, right? So we are located in Kuantan. If you can see, this is the the area within the compound of the university. Okay, so this is where you can have the waterfalls. All right. Now, in Kuantan campus, so this is the faculty. This is my faculty. This is a building where we do lots of activities. So our clinics on the left-hand side. So this is the center will be the administration building. So this is where, if you come to my university or Kulliyyah, so we are going to meet here. Right, so this is how it looks like. It looks like molar teeth, isn't it? All right. At our faculty, we have 6 departments. Okay, we have fundamental, dental, and medical sciences. So this is the department which works almost similar like other faculty of science. Okay, there are biologists, microbiologists, immunologists, biochemists. We have also pathologists, anatomy. We teach anatomy under this department. And then we have pediatric dentistry, dental public health under one department, oral maxillofacial surgery and oral diagnosis, restorative dentistry, prosthodontics, and orthodontics. So we have all 6 departments altogether. And one of the cluster that we have under this faculty is cluster of cancer research IIUM that falls under that is a subset of fundamental dental and medical sciences. So under this cluster, we do lots of research in regards to oral cancer, but we also do some research on machine learning. Okay, other than that, we also do the microbiome study, which many of the activities actually based on the collaboration between our faculty and other university. Now, that's all about the university itself, my faculty. I would like to welcome you guys to come to and join and have a collaboration with us as well. So if you are looking into the application, where to apply on the products that you develop, so that will be an alternative. Okay, so we can help you, we can assist you in regards to the IRAC and so on and so forth. Now let's move forward into the main agenda of the presentation today. We want to talk about the research and development regarding the probiotics. Right. So everyone knows that when we talk about microbiome, this is the center. We're talking about the biological system. So it refers to the entire habitat. Okay, the microorganisms which include the bacteria, archaea, lower and higher eukaryotes and viruses. So it is a composition of all the organisms that living. If we focus on the body itself, everything that lives on our system, in our system, and also on the surface of our skin, they are all considered as microbiome. Now, everyone also knows that the probiotics are actually a good microbiome that resides on the surface of ourselves or even in the gut, right? So in general, we can say that the probiotics is defined as a living microorganisms that provide health and benefit when utilized generally by improving or restoring the oral microbiome. In other words, when we talk about probiotics, it is all about regaining the imbalanced microbiome. So what happened in the gut, what happened in the oral cavity when you have dysbiosis, which is imbalanced microbiome, you want to bring it back, the right or the equilibrium formula for the microbiome in whatever area that you are looking for. So probiotics is one of the methods where you can bring this equilibrium or the balance in the gut, the balance in the oral cavity, the balance on the skin and everything, right? So that is the definition of probiotics. In other words, probiotics actually a living organism, okay, that bring a benefit to the system, either to reduce the inflammation, either to overcome the population of pathogens. So this is the benefit of the probiotics. Now, we know that every one of us might have come across this food. I don't know how many of you have taken lunch, but this is one of very common among the Indonesian. So we have tempeh. If you guys love to eat tempeh, you know it's actually made from probiotics. So we have also tapai. Ni siapa yang duduk kampung tak makan tapai, saya rasa tak betul lah dia. Tak cukup masa lagi lah. So if you are Malaysian, definitely you have taste this. So tapai is actually from fermentation, also have what they call that the probiotics as well as the prebiotic. Now, as I said earlier, most of the time when we talk about probiotics, it is all about gut, right? There is also studies mentioned about in Nature Communication. It says that fasting alters the gut microbiome, reducing blood pressure and body weight in metabolic syndrome patients. So in other words, my brothers and sisters, so we try to, by doing fasting, whether intermittent fasting or whatever fasting it is, so you are actually gaining back the beauty of the balanced microbiome in the gut. So when you have a balanced microbiome in the gut, eventually it actually reduces the blood pressure, it reduces the the body weight and many others. So many other studies also mention that when you have a good balance or a balanced microbiome in the gut, then what will happen is that you will have less stress. So if you are stressed with your publication, you are stressed with your whatever thing it is, okay, looking into nowadays, I just received another email from the university saying that there is a budget cut. And then we have lots of students now struggling because of the, and then one thing as a Deputy Dean, we are looking inshallah, we are going to have 38 convocations sometimes very, very soon, but it has to be cancelled because of PRU. And then there are lots of students asking me, Doctor, we already purchased the ticket, the flight ticket and so on and so forth. I cannot say anything because it's already happened. So Alhamdulillah, today, the reason I asked, I don't remember, Munirah kan? Siapa? Okay, why I choose to be here rather than staying at my university giving talk? Because I cannot focus. There will be lots of students come to my office, even the lecturers come to my office. Hafiz, how are we going to solve this problem? Oh, there are problems with the grouping. The clinic is not running. Many things. So I cannot focus. So that's why the reason I am here, right? So if you look into other perspective in biochemistry, okay, if you still remember the lecture on amino acid pool. So one of the factor when you have increased body weight, you have alkaptonuria and many other diseases because you are stressed. So we want to reduce stress. How are we going to remove or to reduce the stress? So we can also have this kind of methods, which is to improve the balanced microbiome. So now I'm just focusing on something that is very common for many of us. We are talking about probiotics. It always comes into our mind, Yakult, 𝐕𝐞𝐳. So this is the two things that sebut je probiotic, benda dua lah tu lah akan kita masuk dalam pemikiran kita. Am I right? Okay, now I want to change a little bit perspective of you guys. Okay, so rather than focusing on gut, perut aja, now let us talk about probiotics in the oral cavity. Okay, there are lots of questions in the beginning of my career when I started joining. I started my study at the University of Malaya. Okay, in the Faculty of Science doing microbiology, and then I continued my master's in dental science at Faculty of Dentistry, also at the University of Malaya before I joined Melbourne University, together with Azlan and the same faculty but different building definitely. So I learned about probiotics when I joined Melbourne University. It is all about the polymicrobial interactions. Okay, so in the gut, in the oral cavity, we have various of microorganisms, right? We have, you know better than me, you have been doing sequencing and so on and so forth. So we have lots and lots of microorganisms in the oral cavity, in the gut, in oral, in whatever surfaces it is. So now we just focus on a very small area, the biota of the oral cavity itself. So we want to, one of the problems in the oral cavity is because of, you have everyone of the, definitely I would say that 90% of those of you in front of me, you have kids, and then definitely there will be caries in the oral cavity of your children, right? So we cannot run away from it. So one of the factors that lead to this caries development, it's because of the imbalance of microbiome. So you have the factors that lead to caries. Number one will be the sugar, okay, sucrose. Number two, there is thyme. Number three, there will be microorganism. Okay, so these are the three components that leads to the formation of caries. So in other words, if you have thyme, you have bacteria, you have sugar, all these three together, okay, and last but not least, there must be what I call the tooth surface. Kena ada gigi lah, tak ada gigi, tak jadi caries. Again, so you have these four components, okay, you have the gigi, you have teeth, you have thyme, you have what I call that, apa lagi tadi, bacteria, okay, and then you have substrate, which is sucrose most of the time. So you have all these four components, and eventually you can start to have caries, right? So how the reason why caries happens is because of the bacteria has been predominated in the oral cavity. You have lots of acid produced by the lactic acid bacteria. It's on one side it's good, on the other side it's very bad, because this pH, this low pH, eventually will remineralize or demineralize your oral cavity, your teeth, your tooth surface. So sebab tu gigi jadi rongak. Okay, so now with that idea, we know it's because of the microorganism. So we have to bring back the good microorganism, or the microorganisms that can inhibit, or at least to not to kill, but to inhibit the growth of the pathogens of, say for example, in the oral cavity of patients with caries, it is all about Streptococcus mutans. We have Lactobacillus casei. Okay, so we want to control these pathogens. Okay, if we have a very slow or a balanced number of Streptococcus mutans, then there will be no caries. Now, how are we going to bring this what I call balance? By having oral probiotics. That is one of the current trend or current methods that we use in the clinic. It is not very common yet, okay, but that is where we are moving now, okay. So in what I call in 2020, Kleinstein said what a new concept is oral probiotics is a new concept in the prevention and treatment of oral diseases through manipulation of the oral microbiome has been suggested. This is very recent in 2020. Now, when I get into this building, actually I know this place is not today. I met Bazli a few times before. We have students together. I have, other than Azlan, okay, when I come here, the first time I saw mangosteen. Kan, ada dalam gambar mangosteen kat situ. And then just now I saw that you've done on ketum something. So I know that there must be some research you have also been doing on, I assume, on banana. So every one of us know that one of the things that it is very good about banana, it is also known as a prebiotic. There are various types of prebiotic. So what is prebiotic? What we know, probiotic is bacteria itself, which is good. Okay, now what is prebiotic? In a simple way, you can say that the prebiotic is supplement for the probiotic. So bacteria is also consume food, they require nutrients. So the nutrients for the probiotics comes from other sources. One of the sources, a very important source for the probiotic is this prebiotic. So in other words, we can define the prebiotic as a non-digestive food ingredient, okay, that beneficially affects the host by selectively stimulating the growth and activity of one or a limited number of probiotics. So prebiotic is the supplement for the probiotic. Now, how about if you combine both? You have probiotic, you have prebiotic together. So this is what we call as a symbiotic. Okay, so symbiotic is when you have probiotic, okay, the good bacteria, you have the prebiotic. You combine the two together, then you can call them as a symbiotic, right? And then, how about the postbiotic? There are many other biotics nowadays. Previously, we just focused on the probiotic. So nowadays, we are also discussing on the postbiotic. So what is postbiotic? Postbiotic, you can imagine when you are growing bacteria, okay? Many of us are growing bacteria. So you grow bacteria in a media, in a culture media. Normally, we take the bacteria, we centrifuge, okay? We take and then the supernatant, we will discard it, and then you just take the bacteria, isn't it? Right. So the supernatant is actually a postbiotic. Okay, so the supernatant is the postbiotic. If you are growing your probiotic, for example, in 𝐕𝐞𝐳, which is halal, then you can say that I am having, in my lab, I have a postbiotic of what I call a certain bacteria of your interest. Okay, so we do study on postbiotic, and the postbiotic, if you can say that by definition, Salminen said postbiotic is actually a preparation of inanimate microorganisms and their components that confers a health benefit on the host. In other words, if you want to study more on the metabolomics and metabolites, so maybe you can focus on the postbiotic. Okay. Now, it is not just caries. Okay, when you have a probiotic, when you have a balanced microbiome, it is also actually can prevent various of other diseases. So imbalanced oral microbiome have also been reported to cause periodontitis. Periodontitis, Azlan knows more lah. He's been doing on perio when I was in Melbourne together with him. Okay, caries. Okay, it's also in relation with the diabetes, pancreatic cancer. Okay, rheumatoid arthritis, definitely oral squamous cell carcinoma, colorectal cancer, many others. So what I'm trying to tell you is that when you have a balanced oral microbiome, and eventually you can reduce, or at least not to eliminate, not to treat the disease, but to reduce the potential of having certain diseases. Now we are very concerned about SARS-CoV-2, about COVID-19, okay, but not so concerned anymore, okay. But the thing is that when we talk about COVID-19, there is also a very clear relationship between the what I call the oral hygiene and the severity of infection by SARS-CoV-2. So in other words, it is true when Nabi Sallallahu Alaihi Wasallam says, "An-nazafatu min al-iman," what I call the cleanliness is part of faith. Okay, so it means that how beauty is the hygiene, how beauty is when you have a very good oral hygiene, it can prevent various of diseases. So in other words, it's also in, you have to gain a beautiful a balance or microbiome, and eventually you can have, overcoming various of oral diseases, including periodontitis. Now, I want to focus on one of the very serious disease, which is oral cancer. So why is it oral cancer so important? So when we talk about oral cancer, it is actually the number 11. Previously it was number 7. Now it becomes less, but the number of patients been infected, but not infected, that having this oral cancer is still, the number is remain the same, I would say. Okay, no significant decrease. Okay, in 2020, it was reported that the new cases was about 300,000 to 400,000 of new cases. All right. So if you look into this figure, you can see the population of what they call Asia. Okay, in 2020, the mortality reached to 131,000. So you can imagine a very big number of mortality has been reported only in Asia. If you calculate the total number of mortality in 2020, it was 177,000. So a very big number, isn't it? Okay, how about the incidence we have in Asia alone? It is 248,000, 100,000, and the total goes up to 377,000. So you can see that the number is very, very big, Okay, because of that reason, one of the factors that leads to oral cancer, there are various factors. Okay, number one, very common, is because of the oral hygiene is one of the factor. Number two, because of tobacco smoking. Okay, those who are having alcohol also is one of the factor that cause oral cancer. Other than that, because of the infection. Okay, the virus infection. Okay, HPV, for example, and also the latest invention and the latest reports has not so latest. It has been reported since 2016, 2015 that one of the factors that lead to oral cancer is because of the infection by Candida. So those are working on Candida, it's one of the maybe study that you can go about if you want to test whether your extract, whatever thing that you obtain from your study, you want to study if there is any effect on Candida. So you can also relate to oral cancer. All right. Now, not zoom. Okay, it's okay. Now look, this is one of the when I mentioned to you, because of my interest in oral cancer, therefore I look into the study that I studied. I started my journey in 2012 when I was joining Melbourne University, looking into what are the potential pathogens that is in the oral cavity, which I mentioned to you earlier. It's about Candida. So one of the Candida that is very common, previously we studied on Candida albicans. So there are various types of other Candida. We have Candida albicans, we have Candida parapsilosis, cruzi, tropicalis, glabrata. Okay, and then the recent study shows that other types of Candida albicans, other non-albicans, what we call as a non-albicans Candida, has been reported to, to be the isolate was higher. Okay, it's higher nowadays compared to the albicans Candida. So what I'm trying to tell you is that previously maybe we thought that in Candida albicans is the most common isolated from the oral cancer patient. Now it is no more Candida albicans. Okay, it is non-albicans Candida is the most predominant in the oral cavity of oral cancer patient. But definitely the non-albicans Candida include cruzi, parapsilosis, and so on and so forth. Okay, therefore, say for example, if you guys want to do some study on Candida, just nowadays it's not only on Candida albicans. Try to go for other Candida as well. So like what I'm doing with Dr. Bazli, so we are working on Candida albicans. We remove one of the gene. So that one we have done in 2017 until now. How about other species of Candida? Why don't you remove the genes from other species of Candida? Does it going to give the same effect? Does it going to increase the virulence of this microorganism? So these are the study that maybe you can go in the future. So when I'm sharing you this picture, if you look into this picture, it's actually on the left-hand side. Okay, you can see there is a strain. There is a strain, the list of strain ALT1 until ALC4. So ALT stands for Candida, that is from the lab strain, so all the ATCCs. Okay, whereas the ALC1 until ALC4, these are the Candida albicans that are isolated from cancer patient, that are isolated from HIV patient. Okay, so we do study on the two different strains of Candida. Okay, and then when you have this Candida, okay, this is the study on aggregation. Aggregation means what I call how Candida interact with other types of oral pathogens in planktonic. Maksudnya it's not attached to any surface. All right. So when you have Candida together with AN stands for Actinomyces naslundi, SM at the bottom stands for Streptococcus mutans. So when you combine Candida with the what I call bacteria, which with the pathogenic what I call microorganisms, you can see all the red. Red means there is an increase of what I call interaction between microorganism. So when there is interaction, means that this Candida, the pathogens, if you put together with other pathogenic microorganism in the oral cavity, they becomes like very aggressive because they have friends. Okay, so in this study, we just want to identify which one is the friend, which one is the foe. Siapa kawan, siapa lawan. So when you see that some of it is white in color, so meaning that there is no effect. All right. So in other words, okay, you can we can conclude from one of the articles over here. Okay, saying that oral pathogens, they tend to communicate to each other. Okay, to induce disease in the oral cavity. Okay, and then from that study, okay, which we have done in 2012, we work on we make an what I call a conclusion. Okay, that we say this polymicrobial interaction, not yet a conclusion, a hypothesis. We said that this polymicrobial interaction of Candida, okay, with other types of pathogens can induce or have a role in oral carcinogenesis. So meaning that it is not only Candida albicans or Candida cause oral cancer. Okay, it must be with something else, because if it is Candida alone, trust me, in your oral cavity, based on the study, 70% of healthy individual, okay, in the oral cavity must have Candida. Okay, especially if you have caries. So 70% of individual must have Candida. So if you say that all people with Candida albicans will cause cancer, so it is not true. Because you need to have something else to trigger together with Candida, and only then you can have oral cancer. And many other things, many other factors is also involved. If you brush your teeth every day, you keep you ensure your oral cavity is in a very hygiene, so there will be no possibilities. The possibilities of having oral cancer is going to be very, very low. So in this article, we write down a very simple hypothesis discussing on how actually a polymicrobial interaction can be a very crucial factors in causing oral cancer. I will share with you later on these articles. And then in 2020, together with Dr. Bazli, we come across into one other factors. We make into a tiny subset, looking into what will be the gene that is very, very important nowadays in regards to oral cancer. And we found out that ECE1 gene, which is not only for the formation of hyphae, it is also important in inducing inflammation, okay, in what I call that on oral epithelial cell, the normal epithelial cell, and as well as in oral cancer cell line. So what I'm trying to tell you is that this is one of the gene nowadays has been among of the concern, because this gene have only been discovered in early 2015 something. Okay, so if you want to study more, maybe you want to collaborate with us more and Dr. Bazli, so maybe we can go into more detail on the study on this particular gene, Candida lysine ECE1 gene in 2020. Then, many more minutes. Okay, I have 15 more minutes. Okay, now, and then, you know, in the field of dentistry, we always say that Actinomyces naslundi is one of the factors that cause various of diseases, including dental caries. And then we know that that one of the one of actinomyces that is namely Actinomyces israelii cause actinomycosis. Actinomycosis is like those who are, if you can imagine, dia punya bengkak kat sini, and then when you do incision, and there will be lots of pus comes out, right? So that is actinomycosis. So the infection get through the gum, all right? So in the study, we noticed that when we do a systematic review, we found out that, okay, for actinomycosis to happen or actinomyces to infect in individual, it has to be also in polymicrobial infection. So we come into conclusion is that now if you want to induce disease, you cannot just simply make a conclusion this disease is caused by this particular bacteria or this disease is caused by that particular bacteria. Only one bacteria and then you make a conclusion for all disease, right? So that is my what we hypothesize, and we try to confirm in another study. So we make another study. This study is in 2018. Okay, so we do a little bit on study on probiotics and prebiotics. We join together Candida, Streptococcus mutans that cause caries, as well as Actinomyces naslumbrii. And if you can see in this figure, so this red color, this is a figure from Confocal. Okay, we do fish fluorescent in situ hybridization with certain probes and so on. And then we notice that, okay, on the left-hand side, this is Candida. Down here is Streptococcus mutans. Over here is Actinomyces naslumbrii. Okay, at that time, we used a strain that we found out that this strain might be a potential probiotic. We not yet confirmed that this is a probiotic, but we assume it could be a probiotic. So when we combine all these three together, which in this figure D, you can see the blue thingy just, you know, it decreased significantly. The blue thing over here, it's actually the thickness comes from this blue color comes from Streptococcus mutans. But when you combine the three together, you notice that the blue color, it's gone. Okay, it's still there, but it becomes very low. Okay, and then we make a conclusion that when you have these microorganisms, okay, when you have a potential probiotics in a constituent or whatever, you can also assume that in the oral cavity, why some people who do not brush teeth, but still the oral cavity is still good, not so much infection, it's because of there are probiotics. There are good bacteria which balance the other pathogens in the oral cavity. Okay, if you look into the animals like chicken or whatever, chicken doesn't have teeth. Okay, what do you call lembu ke, kucing ke, they don't brush their teeth. But why are their oral cavity, the teeth are very strong? Okay, there are many other factors. Then I would say that one of the factors could be because of there are microorganisms in the oral cavity which actually balance the other types of pathogens. Now, you know, it is not easy to come into conclusion saying that we can use probiotics to overcome oral carcinogenesis, okay, or oral cancer development. It's not easy to convince a dentist, okay, a dental specialist to say, oh, you have to use this extract so that it becomes something that is very good for the oral cavity. You can use that extract. So how many extracts has been we've been conducting study? At my faculty alone, we have been doing on macam-macam lah. Sebutlah extract, whatever extract, habatus sauda lah, extract kayu sugi lah, segala macam extract. But eventually there is no product from kayu sugi, there is no product from habatus sauda, there is no product that comes from our university, my university, I would say that. Okay, so there is a problem with that. Now we want to go in something to one other disease which is very serious disease, which is oral cancer. And how do you convince them? And how do you convince yourself first? So to do that, we started with a systematic review. So we have published in this what I call a Q1 journal in the field of dentistry. So it is archive of oral biology, and we found out that there are microbes that potentially to become what I call a probiotics that can inhibit, okay, that can prevent or at least to prevent oral carcinogenesis, right? I can share with you later. Some of the probiotics include Lactobacillus salivarius and so on and so forth. I'm so sorry because it's a bit tiny again. And now we noticed that when we do some study, we found out that, okay, this what I call bacteria has been studied by others. So what will be the novelty? What I want to explore. So in what I call 2021, I would say 2020, we do some other study. So we combine probiotic, okay, probiotic that we use is based on the literature. We noticed that Streptococcus salivarius K12 has been one of the very newly discovered bacteria that has a potential in inhibiting various of that they claim to prevent halitosis. Halitosis ni mulut berbau busuk. Okay. Then I discussed with my student, why not we do study on this probiotic? Okay. Then we do together with aggregation assay, which I showed to you earlier, and the biofilm study. This is polymicrobial biofilm. Okay. I take Streptococcus salivarius. We add together with Candida albicans and other types of Candida. We look into how does this Streptococcus salivarius actually affect in the biofilm formation as well as the morphology of the bacteria of the Candida. Okay, and then we notice that the number of Candida actually reduced. Okay, when you have Streptococcus mutans when you have Streptococcus salivarius, you combine together with Candida albicans. Then we think that it is not enough. Okay, so what is the difference of our study? If we do the same, people has been doing on Streptococcus salivarius K12, then what is different with our study? Okay, with my study, where is the way forward? Then I come across that, I don't know how many of you like to eat banana. So when I was in Australia, one of the fruit that always with me is banana, pisang. Because it is, you can bring it everywhere you go. Okay, tak payah susah-susah nak basuh, just kupas sahaja. So it is very easy for me. And then I also like to eat banana fritters. So I think, pisang goreng banyak, pisang goreng crispy kat tepi-tepi jalan. And then what people do always, they just throw the skin. They don't use the skin. So at that time, I thought that why not if I take the skin, because it is free of charge, because people just throw it away, and I do a little bit of extraction from that skin, combined together with my probiotic. Maybe the skin possess prebiotic, I don't know, at that time. And then, alhamdulillah, we found out that the prebiotic, in the beginning, we tried to do the study on, panggil apa tu? Susceptibility test. And when you do susceptibility test, the what you call antimicrobial testing, my student was so frustrated. And then when she met me, because we have negative result, there is no result at all. It's no inhibition zone. And then my student comes to me and say, "Oh, doctor, there is no inhibition. There is no inhibition against pathogen." Okay, and then I said to the student, "It's okay. Sometimes it's good to get the negative result." But trust me, at that time I was so scared, because for me, I also want to have a positive result. But I cannot say no to my student. I have to make my student always comfort. And then at that time, I came back home and then I was thinking with my kids, not definitely my kids, my wife. I asked my wife, so how I have been spending this study, and I have been convincing the student, don't worry, but now I'm worried. Okay, so, and then my wife said, why don't you look into some other perspective? My wife is an accountant, so she's nothing to do with scientists. So you ask her about science, she know nothing. No, she's belajar lah science dulu, but when the probiotics so on, but she said that, why don't you look into the other perspective? And then I said, oh yeah, I know this could be a prebiotic, so I combine with probiotic, look into how it grow. Does it makes my probiotic becomes healthier? And then alhamdulillah, we found out that it becomes what we assume, what we expect to get, and then we publish in another Q1 paper in biofouling. So we reported that when you have banana skin together with Streptococcus salivarius, it will further inhibit the pathogens, not only Candida albicans, but also Dubdiniensis, glabrata, cruzi, by more than 96%. See, this is the beauty, mashallah. Something that I assume in the beginning, I thought it's a failed study, but eventually it becomes something that is very, very useful. And from that moment, definitely it's not me, my students was doing this. Okay, we look into the inflammation. Okay, we look in the toxicity study, and we publish another paper this year in another Q1 journal. So we mentioned about, we have probiotics, we have prebiotics, we have symbiotic, we have postbiotic, and all of this has a potential to be used in oral cancer management based on what we have found since 2012 until now. From that failure, it brings us into one of the patent that I want to share with you guys today. We just received this patent in what they call the April, okay, granted in April, 26 April 2022. What I'm trying to tell you, if there is any students here, don't worry if you have failed, you have negative result, because sometimes your negative result will bring you, I don't know, pattern. Even your lecturer might not have the pattern. Okay, so this is the thing, the idea that I would like to bring to you guys. Now, from that failure story as well, it brings me into commercialization. So this is the product, this is the company that I work currently with, Jardin Pharma Berhad. Okay, we have an MOU, an MOA together, both MOU and MOA. So we have been reported in what I call, I don't understand a single word except Jardin Pharma in this Chinese paper, but at least I can show you it goes everywhere. And then, you know, with this failed result as well, we managed to get into commercialization. So the company has been building their own websites and so on and so forth. So they do commercialization. If you can, you want to purchase this, it's 19 ringgit now. It's you just Google habitus.com and you can have this 2 toothpaste, which is actually based on our failed study. Okay, so we have the one on the left. If you can see, natural symbiotic toothpaste. So this is the first, the second failed study. Okay, so we do study for the children and also for the adults, all right? And then another product from that field study as well, we managed to have our first natural symbiotic mouthwash. So out of nowhere, okay, from sampah yang orang buang free of charge. And alhamdulillah, from that field study as well, we managed to have our first startup company. We run together with Jardin Pharma Berhad. So we have our first Jardin Probiotics Research Center. So we do lots of studies on probiotic because we know that our university is not an RU, so we don't have fund enough for us to set up a center. So we work together with the company. We have an MOA. We ask the company, come on, help us to provide a laboratory, and we give the idea. Okay, if I want to travel, then you pay everything for me lah. So this is located in Kedah. My kampung is in Kedah. So every time I go back to my kampung, so I don't have to, tak fikir duit lah. Okay, because everything will be sponsored. And then if I want to ask for leave, I said I have what I call a meeting with Jardin Pharma Berhad, so I can still save my annual leave. Right. What I'm trying to tell you is that, okay, do not worry when you get a failed result. Okay, as long as you have integrity, you know that your result is, if it is failed, just report it is failed. Okay, and many things will come with you if you are amanah. Okay, you are sincere, but don't be, you have to think twice lah. So for me, I have my own reason why I publish the failed or the negative result. So because I want to support my symbiotic, right? So now I heard that Azlan told me when I discuss about commercialization, maybe something that is very useful for you guys. Okay, so I have come across in my study that lead to commercialization, we have several challenges. Number one, the challenge I have so far, when we talk about 2 minutes. Okay, so the first challenge is the industry itself. So because, when I have this symbiotic, the idea, I already filed for patent. So when you approach a company, okay, sometimes you approach big company, big pharma. You go, for me, I go for Colgate lah, macam-macam lah. People they don't listen to us. Okay, they don't care. Who are you? I'm not a dentist. I am a microbiologist. And why should I trust you? Because those who do research is us. We are not a clinician, but we have the knowledge, the information that the clinician gave to us. So to overcome this, you have to work with the clinician. So that is why I open for you guys. If you want to do collaboration, I don't know, maybe it is easy to collaborate with your university, but our faculty, we open to everyone. So that's why I have collaborators, not only from UPM, also I have collaborators from other university. Okay, so I would say that if you want to move forward, but again, another problems that we have is the regulatory. There are various understanding about this regulatory. For example, if we have patent, I and Bazli, we work together, and then there are, how are we going to distribute the patent? Is it 50-50? Is it 30-70? So that is how you deal with the university. So the university should help us because we do the study, but we are not trained to, you know, to do this commercialization. Our PhD is not commercialization. Our PhD is, I don't know, biochemistry, microbiology, whatever you are. Okay, so another perspective of regulatory is that when you want to produce some products, so you have to follow the guideline by the Ministry of Health. Like you have to deal with NPRA, if it is falls under pharmaceutical, what will be the rules and guidelines do I have to follow? And then another problems that I found out that the biggest challenge that I can see here is we tend to work in silo. So I don't know you guys, but at my university, we thought that this one has to be belongs to UIA. Because if we pass to UKM, then UKM will get something, and then it's going to be very, so we have this kind of perception. So if we still have this silo punya work, then I think we are not going anywhere. I don't know how long has INBIOSIS been introduced. UIA has been there about 40 years, so we are celebrating the 40th anniversary. So if you think about the products that has been materialized from our study, then you know that there are some of the problems happening there. Okay, so one of the things that I come across, I look into the problems comparing to my experience when I did my PhD is that we tend to work in silo and we are not confident with our own product. Okay, we do lots of studies, but we don't, when we want to pitch and then we pitch and then we are, if you ask me, did I use my own toothpaste? I must use my own toothpaste. Sometimes we produce something, but we use some others product. Because of what? We didn't trust our own product. Okay. Last but not least is because of the raw material processing. Because imagine banana skin is not easy to get, isn't it? You have to have how many farmers. But don't worry, there are lots of what I call outside there. I don't want to say which country they are producing it. So you just get it from what I call, you can just import. And before that, I would like to thank my students, my lab mates, what I call, all of this down there is my students. Dekat rumah saya, lepas makan-makan, kenduri sikit hari tu, kenduri lah, lebih kurang. Again, so I would like to acknowledge all my colleagues, members from all these universities. And with that, thanks to your, what I call, thanks for listening to my presentation. So if you want to contact me, so this is my contact. You can have my phone number, and everything is over there. Okay, so with that, I end my presentation. Thank you very much for your attention. Thanks for listening to my presentation. So if you want to contact me, so this is my contact. You can have my phone number, and everything is over there. Okay, so with that, I end my presentation. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Hafiz. Thank you, Dr. Hafiz, for the interesting topic. So now I would like to open the floor for questions. Yes, any questions from the chat box? So just to get the ball rolling, maybe I start with one question first. So we know that the oral microbiome is a very complex environment, right? I think there's a few hundred species. I think I forgot from my PhD work. So just wondering how when you try to look for a new symbiotic, for example, right? How do we measure the effectiveness in a large scale? Because you've shown results from a lab scale, right? But for you to create a product, it must be safe, must be compatible with everyone and so on, right? So any comments on that?
All right, to answer to that question, so that is why you have to follow the guideline by NPRA. Okay, not everything has to go to preclinical testing or even to clinical testing. Okay, so there are some products which belongs to certain categories. You have to go into the guideline by inquiry. So if you want, that is the thing now as a scientist, we tend to go for FRGS and then PRGS and then mostly grant, lepas tu tak dapat, ulang balik, and then you keep on do the cycle. Okay, and then the product will go in nowhere, and then you know after sometimes the product has been produced by other countries. Do you agree with me? Okay, so how you can do this quickly is you have to tag with the industry, because the industry knows what are the markets, what are their target, what they want to focus on, because we are lack of manufacturing, we don't have kilang pun. Kan? So we are lecturer, we are academician, we do research, we don't do marketing. If we have our own company, then we'll be kicked out from the university, because that is the guidelines by the university. We have signed everything. Now, if we want to go for clinical trials, it's true, it depends on what field or what products are you producing. If it is fall under pharmaceutical, then you follow the pharmaceutical guidelines. We do preclinical testing, but in our PRGS. Okay, we have PRGS, we just closed the PRGS. Okay, so in that PRGS, we do preclinical testing on rats. Okay, and then we do clinical testing. It is not a clinical testing, because if you do clinical testing, then there will be some things that you have to follow later on. But we give the product to the patient, to our customer, and the customer give us the feedback. Okay, we don't claim that as a clinical test, clinical trials, because that is not a clinical trials. Okay, if you can imagine, there are lots of product. Azlan, kau ingat, kalau ingat masa kita dekat Melbourne dulu, dekat produk tu dia tulis no animal testing. Okay, so siap tulis no animal testing, how can it comes out and sell at sold at dekat mana kita duk beli selalu? Chemist, warehouse, tempat-tempat murah lah kalau siapa pernah dengar. Macam Guardian lah kita. So how can that get into the chemist, pharmacy, if it is not accredited, it is not certified by ministry. So as long as it follows the guidelines, if you want to produce product, so make sure you read the guidelines. I don't read the guidelines because I don't know. I'm not a pharmacist background pun. And therefore I tag along with many collaborators. I have in my team, I have, if you can, when Azlan read my CV just now. So it is all about collaboration. I have a very limited manpower. I have a very limited knowledge. I don't have many things. My laboratory is very, I mean, not so bad lah. I mean, at least I can run something. But if you want to do something, you have to go from the back, look at the back, what is the end product? Okay, and then from that you study what are the things that you need, and then you start to do the real study. That is how I work with lah. So if you want to prepare a product, so look into the guideline. If the guideline says you have to do clinical trials, then clinical trial it is. If not, then it will fall. If it is falls under the pharmaceutical, then you follow the pharmaceutical punya guidelines. Okay, so inshallah the rest will be as smooth as possible lah. But I cannot promise it's going to be smooth, but at least you know what will happen if you do certain things.
So we have one question from Dr. Go. What's your greatest challenge to bring the product to market? How do you validate the efficacy of the toothpaste as claimed?
All right, thank you, Prof. Go. Okay. Right. So the question is, what's your greatest challenge to bring product to market? Okay, my greatest challenge is, you know, when you have a patent, okay, when you have a patent, then the company want to buy the patent. Okay, normally the industry, I don't know with you guys, I have my own experience, you might have yours. Okay, so my experience, the challenge I have is not to have to get the grant. FRGS, PRGS, whatever grant, the MOSTI grant, I have been turned out many times with the MOSTI grant, but it's fine, it's okay, right? Okay, now we noticed that from my experience, when we have a patent, when you want to sell the patent, sometimes the university will put a very, you know, the amount is something that may be beyond the expectation of the industry, except if you sell it to like a very big company, Amaniaga ke apa benda ke, those who are very kaya lah. Okay, but if you want to sell your patent to a local company, okay, sometimes it's going to be very hard. So they can offer you certain amount, certain budget from them. Okay, But then it will be with our university whether they want to approve or not. So my challenge that I get at that time was to convince the company that we can do what I call that, we can have this outright sale. Okay, kita boleh jual patent kita, but it has to be at a certain amount that is acceptable by the university. Then what did the university do? For IIUM, we hire the third party to do patent evaluation. Okay, so that takes like 1 year before I can, eventually get this product approved by the university to be marketed and to be commercialized under Jadin Pharma Berhad at that time. So Prof Goh, that is my challenge. We have patent, but whether the university are willing to sell the patent at what price, whether this price is affordable to the company or not. So that is my challenge. And when how do you validate the efficiency? Okay, so for the validation, we have various of methods in validation. We do preclinical testing on animals. Okay, other than that, we definitely we have various of in vitro study. Okay, to study the efficiency. We do also study on what I call that. We also do the study on the stability and many others. So one thing what is so unique about what we have been doing is that we have this symbiotic. So this symbiotic is actually a combination of pro and prebiotics. Okay, it is like if you know chlorhexidine, it is not a substitute of chlorhexidine, but you can say that it is another chemical product. It's not a chemical definitely, but you can assume that it's a product. Okay, a chemical. Okay, so you just add on this chemical into the pre-formulated toothpaste. The toothpaste is very common. When you ask to do a toothpaste, it's a very common. Toothpaste buat macam mana? Tambah-tambah A, B, C, D. The active ingredient is the one that we aim, okay? That we add into the toothpaste. It's not about the formulation, how we do the toothpaste. We add the symbiotic formulation inside the toothpaste formulation. So everyone knows that toothpaste is good. You brush your teeth, it's going to clean your teeth, okay? But the thing that we add, we just make it something that is useful, okay? Only that bit. And how do we confirm? We do some testing. Definitely, we make some survey, ask the users, is there any difference, and so on and so forth. So from that, we confirm that our product is efficient. Okay, but there is still need more study. Definitely, we cannot conclude there because we are scientists, right? We cannot just simply say, oh, this is good for everything, because our main objective is to reduce the potential of oral cancer. But for that, for this time being, we still cannot claim that ours can prevent or reduce the potential of someone's having oral cancer, or this toothpaste can be used as oral cancer management. But for the time being, we can say that our product has certain benefit based on what we have been trying or been doing in the lab, as whether it's a preclinical trials or our in vitro study. I hope that answers your question, Prof.
My question is, thank you for very interesting. My question is, you are using this banana peel, isn't it? As we peel the banana, it's very easy to get molded. How do you preserve the banana peel itself before you can extract it and make it into formulation and all? Thank you.
That is a very tricky question. Okay, now you can imagine we are not producing only one tube of toothpaste. One batch will be like 1,000, 2,000. So how many banana peel you are going to get? How are you going to extract all this? Do you have enough, what do you call that, HPLC, bukan? Apa nama, extractor nak buat? Okay, if it is water-based, even, is it going to be different species? Pisang berangan ke, nak pakai pisang emas ke, nak pakai pisang awak ke, nak pakai pisang abu ke? We have lots of pisang, kan? Okay, now, that is why I said earlier, when it comes to fundamental study, fundamental research, PRJS, definitely we are going to make everything under control according to what we proposed in the proposal. But when it comes to the reality, when you want to do the production, it is impossible for you to do the extraction yourself in your laboratory. The reason is you want to sell it very, very cheap. If you sell the toothpaste at RM35, siapa angkat tangan nak beli ubat gigi RM35? Tak ada. So you want to make it as cheap as possible. So we import from outside lah, the extract. So the people out there, they already have what they call, you just Google lah, kat Alibaba ke mana ada hala ke, there are various of extract. So you can ask from them the certificate, everything. Okay, you can ask from them halal, sijil halal, semua ada. Okay, then you bring it in, you do the testing. Does it give the same result as what you have in when you did your fundamental and the preclinical trial? If it gives the same result, then you produce with that particular extract. Okay, so do not, if INBIOSIS are able to have a very large scale punya production kilang nak buat extract, then insyaAllah I will buy from you guys. But if not, we just import it for the time being. Yeah, hope that answer your question.
Is not an issue. Just buy the extract. So I guess yeah.
For the product that put in next. Who do the sell or should that be also?
Okay. To answer to that question, we have an experience previously. I worked with another company, not Jadin Pharma Berhad. So I hope maybe this is another what I call challenge that I have with my what I call product. Okay. So in I think 2019 ke 2018 kot. So at that time there was a grant, they call it as ICF, International Collaboration Fund. Okay, 2019, okay, under Ministry of Science and Technology. So at that time, I was given an instruction by the Deputy Rector, if I'm not mistaken, to apply for this grant, International Collaboration Fund. And at that time, what do you call that? I was looking for a company because I still want to have a company to run together in the grant application. Fine. And then I have this company, we have been doing lots of things. I have been visiting their place, and eventually we come to a conclusion that we can do together, but for their party, they can only do like marketing and so on. But in terms of production, or in terms of production, yes, they don't have a GMP factory even. So when we do presenting for the interview, evaluation, we got rejected because of one of the factor, because the company does not have what I call source, or they are not able to support us in terms of what I call producing the bacteria itself and many others. And then I started to find out other company. Okay, and then Alhamdulillah at that time we have Jadin Pharma Berhad. And then another reason of rejection of my first time apply the grant, saying that I have patent, but my patent is not yet commercialized. And in the next cycle, there is no longer ICF, but they call it as TED1, Technology Development 1 grant something. And then at that time, I look into a company which are able to help me in producing the bacteria in large scale. Okay, because of the one of the rejection earlier, not only on marketing, on the commercialization, but also the contribution to the company for the grant. Say for example, if this grant is rejected, will the company support us okay, to run the study, right? And then what happened then? I found that this company, okay, we do definitely RMC is assisting us. So together with RMC, we found out that the company are very, very interested in probiotics. And we applied the grant together. And before we applied the grant, we said, okay, why not if you guys help us by providing a bioreactor? Okay, so that we can say that you are very supportive. And you guys, why don't you, commercial our product? It's not a product, it's just a market testing. And then they agree to that. Okay, so we don't have any problem with that. We have our, what we call a very big 10 liter punya bioreactor. So we produce the because to produce 1,000 toothpaste, you don't need that 10 liter punya bacteria. It's a very small amount of bacteria for each panggil apa tu, tube. So the production, because in the beginning, they didn't have the expert, they don't have the microbiologist. And I advised the company to get a microbiologist. And then they follow. They get a microbiologist with PhD. And then from that, I said to the company, why not if we have what I call a factory that that produce probiotics, not only for you, but also you can commercialize to other company that require probiotic. And from that, we start to have our first startup company, which is on the production of probiotics. So the question you asked, Dr. Bazli, are they the one who produce the probiotics? Yes, they produce the probiotics, but under our advice, we advise them, what should they do? What will be the ingredients that you should do? How do you manage? How do you manipulate the pH? What is the temperature? What is the rotation punya ni? Many things. So they do the production. For us, we do the advice. Okay. So yeah, so far that is what we have been doing for the current production of the toothpaste and many other products based on the probiotics.
Yeah. Your case, you also are responsible for this.
Okay, let me answer to this question. Okay, one last question. So here we have, which one should be done first, patent or commercialize? Just curious how good the bacteria survive, I would say that, in toothpaste. Okay, so that is a very good question. Now, in the I will combine these two questions together. Okay, so which one should be done first, patent or commercialize? So I would say we go for patent first. Okay, because if you have patent, then you can protect whatever thing you want to do around the patent. Okay, if you already commercialize but you don't patent, you don't have any copyright, even you don't IP it, then people can copy your thing and then they claim that is theirs. Okay, so you go for patent first, or at least an IP, whatever IP, it's fair enough. Whether it's apa nama trademark ke, trade secret ke, as long as it is been IP. Okay, so just curious how good the bacteria survive in toothpaste. Yeah, so that is the thing that we are. We have PRGS. Okay, so in that PRGS, we do stability tests. We do assess the efficacy test. So we put the bacteria in the toothpaste formulation. So we have to remove certain things. We have to play around with the formulation. So looking into what can kill the bacteria, then we have to remove the bacteria. So the latest technology that we incorporated into the toothpaste is that we encapsulate it in a micro capsule. So this is another technology which is actually I have been doing during my PhD. So we encapsulate the bacteria in what we call in the bits. Okay, a micro capsule bits. You cannot see them. Okay, so we encapsulate so that there is no penetration from fluoride that can kill the bacteria. So that is the technology we incorporate. It's not like a simple toothpaste. Okay, so a little bit complex, but not so complex, right? So I hope that answer your question. We have been testing. We have tested the efficacy. We have tested the stability for like a period of three months, and then for this 3 months is actually equivalent to certain years. I don't remember people from pharmaceutical, they are very expert on this.
Thank you, Dr. Hafiz. So I guess I'll end the webinar here. So I'd like to thank Dr. Hafiz for the very interesting topic.